Good. Well, welcome everybody to this month's ISFE webinar entitled Plant and Algae Protein Sources and High Pressure, Potential Applications and Limitations by Avi Shpigelman. Associate Professor Avi Shpigelman is the head of the Laboratory for Novel Food and Bioprocessing in the Faculty of Biotechnology and Food Engineering at Technion in Haifa, Israel. Avi completed all of his degrees in the Technion, followed by a postdoctoral stay in KU Leuven in Belgium. Avi's research focuses on the complex interplay between processing, the structure of food components, their behavior during the shelf life of the product, and finally, their bioactivity in our body. The idea that drives the research is that the design of food processing should carefully balance all various aspects necessary to our health and well-being. Currently, the main areas of activity are non-thermal processing, novel protein sources, and polyphenolic compounds. The research strongly focuses on the impact of high-pressure processing as an alternative non-thermal technology beyond microbial inactivation. Avi also serves as an interim head of the Technion Sustainable Protein Research Center and serves as the Vice Dean of Graduate Studies in the Faculty of Biotechnology and Food Engineering. So before we start, please remember to mute your microphone during the presentation, and if you have any questions, you can write them on the chat so that you do not forget them, or you can also ask them directly to our speaker at the end. I can also read them aloud for you. So again, thank you, all of you, for being here. Welcome, Abby, and thank you so much for being our today's speaker. So now the audience is yours. Thank you so much.
Thank you very much, Viri. Thank you very much, Viri and Jorge, for the invitation. I am very happy to be here. Viri, just to make sure everything is clear and seen, everything is okay?
Yes, sorry.
Okay, good. So today I will talk about two of my passions in research, and it is on the one hand, high pressure processing, and on the other hand, alternative protein sources. That is currently actually is a topic that gaining a lot of interest and a lot of traction all over the world. So I will start with the most important thing, and it is to say thanks for all the students. I will show some results from several projects that we have been doing in our lab, and it's important to say thanks to the students that are actually doing the hard work. So thank you to all of them, students in my lab. So let's start with why are we looking at alternative protein sources? So it's quite well known that in the current way that we are producing our food, we are at the same time also harming, and some might say even destroying our planet. We can look at it from the terms of the greenhouse house emissions. We can talk about it in terms of health of our own health, and we can also think about it in terms of the quality of life of the animal that we are using as sources for our protein. If we look at all of those together, it is quite clear that the current way that we are producing food, and especially the current sources of protein in our diet, is not sustainable and very possibly even unhealthy for our future. So this point has been gaining a lot of interest, and many governments and companies and also in the academic world are trying to look at new ways to solve the protein problem. And there are different types of suggested solutions. Some of them are talking about growing our protein in a bioreactor, so the cultured meat solutions. Some of them are talking about using insects as the protein source, so little animal farming. Some of them are actually talking about different types of bioreactors that grow microorganisms inside that will be the protein source. But currently, and possibly also for the upcoming future, the most likely protein sources are protein sources that are based on plant food, and some also consider algae, although they are not plant or not exactly plants, but being part of the same subgroup. Okay, now let's talk a little bit about pressure and about pressure in the food industry. So pressure is an important thermodynamic parameter that is common in a range of different technologies that we are using to manipulate our food, to manipulate the structure of our food, and also to ensure microbial safety of our foods. And we can see that some pressures are relatively low, although above the atmospheric pressures, as you can see here on the left, and some pressures that are used are quite high, and we are talking about hundreds of megapascals, as you can see it on your right. Both can significantly affect the structure of protein. It's important to say that probably the most well-known technology currently used to manipulate structure of plant-based protein sources is the extrusion. But today I will talk about a completely different story, and it is the effect of hydrostatic high pressure. This is the region in terms of pressure and temperature, and how it affects plant protein sources, and why it can bring us a potential benefit. Just really, I'm not going to go too deep into what is high pressure processing, but several important aspects that are also important for the rest of the presentation. The first one is the fact that high pressure processing is one of its fundamental is the Le Chatelier principle, and the Chatelier principle suggests that upon application of pressure, reactions that result in a lower volume will be the preferred one, or structures. We also have the isostatic rule. That means that pressure is transmitted all over the product, or over whatever we are putting inside the pressure vessel instantaneously. Mostly, this results that covalent bonds are mostly unaffected, but on the other hand, ionic interaction, hydrogen interaction, hydrophobic interactions are strongly affected. On the one hand, it can have a very complex effect on enzyme activity, and as we will later on see, it will also have a significant potential effect on non-enzyme protein structures. In addition, we also have the adiabatic heating, so a small temperature increase due to pressurization. And a point that is often overlooked, but might be quite important, and we will also see it in today's presentation, is the fact that because ionic interactions are affected by pressure, we also observe pH changes during pressurization. Here on the right, you have a nice image created by AI, so thank you, ChatGPT. And the fact that 3 elephants are in the image there is a reason for it, and the reason is that the pressure formed by hydrostatic high pressure processing used for microbial inactivation is approximately equal to 3 elephants on top of each other, standing on a coin with a surface area of about a dime. So why plant protein and pressure, and why now? So we see an increasing trend in the research of plant proteins on the one hand, and there is also interest in a great range of plant-based protein sources. You can see it here on the right. It includes protein sources from cereal and from legumes. Actually, the main one currently that are being studied are the ones from legumes, but also from oil seeds and nuts, and even from tubers like potato. And actually, this will be one of the main stars in today's talk. When we are talking about replacing animal-based proteins with plant-based proteins, that's not always an easy task of just, okay, I will take this protein and replace it with the other. Plant-based proteins often suffer from a various limitation. Some of them are related to the nutrition that we will not talk about a little bit about this a lot today. Some also suffer from undesirable flavors. And again, this is not the topic of today's talk, but they often suffer from poor technofunctionality. It's related in many cases to their poor solubility or relatively poor solubility. It is, of course, based on the fact that many of the plant and algae proteins are not designed to behave as neither dairy or meat proteins, and very often they suffer from relatively poor emulsifying, foaming, or gelling activity. And that leads us to the main topic of today's presentation, when we will have some examples and some, I believe, to be interesting information from the literature about the effect that pressure, hydrostatic pressure, has on plant and algae-based proteins, and what potential advantage it can bring to the table, and still what are the limitations that we see and gaps in the research that hopefully we can fill together. I will start with really short terminology because very often different terms are being miscorrectly used by people, and it is important to have the same box of terminology so everything will be clearer. So first is the protein denaturation. So we consider protein denaturation is any modification of the secondary, tertiary, or quaternary conformation, but not related to rupture of peptide bonds. Association is some changes occurring in the molecular level. So for example, like dimerization, and those are related to quite weak bonds at specific interaction sites. The term aggregation or coagulation or precipitation is actually used to define protein-protein interaction that result in the formation of large complexes with high molecular weights. And gelation, the last step, is an orderly aggregation that results in the formation of three-dimensional network based on protein-protein, in this case, interaction. Very often when we are talking about food, we are talking about hydrogels. So water is trapped within this three-dimensional network. When we are talking about the correlation between pressure and proteins, then there are some well-known studies. For example, here on the right, you can see a very interesting phenomena that what happens when we are either heating or pressurizing a globular protein. Very often it is considered as a general for all proteins, but actually this is more accurate to present this about globular proteins. And we see, of course, that if we are increasing temperature at some point of time at atmospheric pressure, we will reach heat-induced denaturation. But this can also happen by the application of pressure. So at the same temperature, for example, at the room temperature, if we increase pressure. At some point of time, we will have pressure-induced denaturation. But what is interesting that if we combine an increase in both, so both increase in pressure and increase in temperature, that the point at which we will have the denaturation will actually be higher in terms of temperature than the point of the denaturation at atmospheric pressure. Also, we should keep in mind that different pressure levels result on different effects on protein structure. If we are talking about relatively low pressures below 200 megapascals, but it is quite a fluid definition, we are usually talking about disaggregation, so breakage of large protein complexes, while higher pressures, especially if we are talking about pressures that are above 400 megapascals, we will get complete unfolding that can be partially reversible, but it can also be non-reversible. And this kind of an unfolding, the non-reversible one, might at the end result in gelation. And the unfolding will usually result in the exposure of the more inner parts of the protein structure. And those parts are usually parts that are rich in hydrophobic amino acids. And at the end, we can also have this complex aggregation or gelation. So first example is what happens to potato protein upon application of pressure, and we wish to compare pressure to heat gelation of potato protein isolate. And the next step, we wanted to identify if there are differences and what are the reasons for the difference. And the last point is the applicative point, when we ask ourselves what advantage can the pressure-induced gelation can bring to the applicative table. So first, let's look at a very simple test of the spectroscopic changes during pressurization. So in this case, we are not measuring the before or after. We have an in situ spectrophotometer that allows us to study in the UV region the effect of pressure. And when we are talking about atmospheric pressure, because we were working at a very low concentration of protein and the concentration that should not, in theory, result in aggregation, and you will see the theory is not actually fully correct. We did not see a significant effect of temperature, just a small effect that you can see here from the purple to the other lines that is probably related to a better solubilization by some heating. On the right, you can see here that in our isolate, the potato protein isolate, the main protein is the patatin, which is what we have expected, and it is a relatively soluble protein, especially in the terms of the plant world. But we also see here a high molecular weight fraction, and it is of importance in Okay, now we will look what happens upon pressurization. So the pressurization was done at a constant temperature of 40 degrees C, because on our next steps, we will use this temperature as the highest temperature reached during the adiabatic heating on a different high pressure system. And here you can see that we do see a change at the of the UV spectrum at the region typical of proteins. We see a smaller change around pH 3 and a very significant change at pH 7. The change in the spectrum as a function of pressure at pH 3 is partially related to the fact that upon pressurization, the effective concentration is increasing because the volume is decreasing. But it might also be related to small changes in the structure of the protein and the beginning of some exposure of some specific domains. On the other hand, the change in the spectra at pH 7 is more complex, is much more significant, and is likely related to some initial aggregation or dimerization of the protein, and we will explain it soon. Later on, we increased the concentration to concentrations that are above the gelling concentration needed for this isolate, and studied the rheological profile, so the storage and loss modulus is a function of angular frequency at temperature and at different temperatures and after different pressures, all treated for half an hour because we wanted to make sure that the gel received enough energy. So you can see here that when we look at the thermal gels, already at temperatures higher than 45 degrees C, we observe quite a significant storage modulus, and I can also let you know that all over the frequency range, the storage modulus was higher than the loss modulus, so it is clearly a physical gel. And you can also see that above 60 degrees C, there is no further increase in the storage modulus. Okay, now the interesting point. What happens upon application of pressure? So upon application of pressure, at the two conditions, we saw a different phenomenon. At the pH of 7, we saw the formation of weak and fragile gels, yet, of course, they were still gels. G prime was higher than G double prime, but we could not fit a power law to those gels because they were very fragile. On the other hand, at the pH of 3, at 300, 400, and 500 megapascals, pressures, gels based on pressurization were formed, and their storage modulus was quite similar at the pH 3. At 500 was quite similar to the storage modulus of the gels at 55 degrees C made by heat. And another potentially important observation that we have made, that we do not still currently have a full understanding of it, is the fact that if we started the pressurization at quite low temperatures, so 10 degrees C approximately, we could not get any gel at any of the pressures. So there seems to be some contribution to the temperature, to the adiabatic heating, to the formation of the gels, although our maximum temperature that we have reached during this treatment, which was just below 40 degrees C for a very short point of time, is not a temperature that allowed thermally induced gelation. And this outcome is quite interesting because it is different from an observation made more than 100 years ago by Bridgman, who worked on animal-based proteins, egg and milk, and he suggested that the effect of temperature, while was not large, seemed to be such that the ease of coagulation increases at the low temperatures. contrary to what one might expect. And this complex behavior, the contribution of temperature, of changes in temperature to pressure-induced gelation was already seen several times in the past for animal-based proteins, but currently we do not have a comprehensive understanding of the reasoning for this. And that's something that I still hope to better resolve in the future. Now let's go back to the pH shifts. I mentioned that I believe that they are quite important and then can help us understand the phenomena that we observe. It is well known that pH of solutions under pressure changes. It is correct for both food products and also for buffered solution. And here on the right, this was even correct for a simple model system that has only beta-lactoglobulin inside water. In this case, and unfortunately, as I will show later, we did not fully comprehend the importance of this phenomena to the protein gelation. So we have performed the study in a pH 7 phosphate and pH 3 phosphate buffers that we know we can calculate that have a significant change in their pH upon pressurization. Another important point to keep in mind is the fact that plant proteins have a complex and varying profile of protein solubility as a function of pH. You can see here some results about a range of lentils, beans, and so on, legumes. And you can also see here on the right that at the end, it's also dependent on which specific protein we are talking about. So if, for example, the albumines in sunflower are quite soluble all over the pH range, while the globulins are not. So I'm taking you now back to the UV absorbance spectra that I've shown a few minutes ago. We can see here this aggregation. This aggregation is probably related to the fact that upon pressurization, we are getting closer to the isoelectric point of the protein. And it is known that that even for thermally induced gels, when we are getting closer to the isoelectric point, poorer gels are formed, and very often we are only obtaining aggregates of protein that are unable to form a clear and 3D network. So we believe that this pH change is the reason that our gels at pH 7 were significantly weaker than the gels obtained at pH 3. Another point to keep in mind that I'm not showing the pressure of 500 because at 500 we did not get any gels at all. And this can also support our conclusion because of the fact that the higher the pressure, the stronger the effect is expected to be on the pH level. Building on this understanding, we actually switched a little bit the topic and started to look also on the kinetic of changes of polyphenolic compounds, so degradation, not related to protein, but the moment we switched from working in buffered solutions and phosphate buffer solution to specifically oriented pressure stable buffer, we could verify and measure accurately the activation volumes of a range of of the degradation in a range of polyphenolic compounds, allowing us to reach quite clear conclusions about the effect of pressure separated from the effect of pH on the degradation kinetic of those compounds. And we actually want also to take it back to the protein and verify our conclusions in the protein. When we are talking about the properties of such gels, so we could easily see, and I will step up the speed a little bit because time is running short, we could easily see that the hardness of thermally induced gels is better. The water holding capacity of the thermally induced gels is better, and it is a very clear sign to the fact that the network, the 3D network formed by pressure-induced and thermally induced gelation is quite different. When we looked at the proteins that are released from those gels that we tested for the water holding capacity, we could easily see a potential explanation for this phenomena. Here on the right, you can see the thermally induced gels, and you can see that the high molecular weight fraction of the plant protein isolate is quite clear after the thermal gelation. So those proteins are likely strongly contributing to the network formed, to the gel network formed by heating. On the other hand, upon pressure treatment, we still clearly see the bands of this high molecular weight protein, possibly explaining the fact that we have a lower strength of those gels. And you can also see that patatin, while involved likely in the network, is not the main protein contributing to this network formation. So the last step at this was to see, okay, why can this protein-assisted gelation can be useful? So for example, it can be useful if we are talking about gels that contain thermally liable compounds. We use a model compound, ascorbic acid, and we could of course easily see that for gels having a similar textural profile, so 500 megapascals at pH 3 and 660 degrees C also in pH 3. So we can clearly see that the concentration of the remaining ascorbic acid after gelation is quite different, with a great advantage for the thermally induced gels. The next important step is, of course, understanding the digestibility of those systems, and I will not go in depth into this topic, but we could see that in many cases, the digestibility of the proteins when treated by pressure is similar to the digestibility of proteins from the same sources treated by heat. High pressure can also be a pre-treatment before thermal gelation, which is also interesting. And you can see here on the left peanut proteins, and you can see here that upon pressurization to 100 megapascals, there was a significant increase in the hardness of the later formed thermal gels, and this was also correct for wheat gel, wheat protein gel. So, but the pressure, the optimal pressure was quite different. So we have the pressure-induced denaturation can allow a later harder thermal gel, but the optimal pressure is of course source dependent and likely also dependent on additional external factors. Others have also suggested high pressure as either a pre-treatment or as a treatment together with an enzymatic activity at relatively low pressures of 100 and 200 megapascals, and it was suggested that it can improve the hydrolysis of the protein by enzymes. Whether such an activity can be economically viable, that's a different question, but in terms of the effect itself, it was quite clear. Next example is pigments that are connected covalently to protein. And this is the case, for example, for the blue color of the pigment of spirulina. So if we look at the blue color of the pigment of spirulina, it is well known that it is not stable to processing. There is a significant color loss upon pressure, upon thermal treatment, and some also have showed that this is also the case in terms of pressure. But we wanted to have a deeper look at this phenomena, so we looked at the UV absorbance. This is an easy way to monitor color upon thermal treatment. Here you can see it on the left, and pressurization for a very long period, 20 minutes. Again, this is just model system. We wanted to be clear about the effect we observed at different pressure levels, and we saw a similar phenomena, although the effect upon thermal treatment was significantly larger, but we can see here the decrease in the absorbance in the visible range, and it is correlated with the loss of the blue color. But when we were trying to quantify the kinetics of the color loss by monitoring again the absorbance at 620 nanometers continuously using our in situ spectroscopic system, then we could see and quantify the color deterioration rate. And based on this color deterioration rate, we can conclude that indeed we see a clear negative effect of pressure and temperature on the stability of the color, but the extent of the effect is lower for the effect of pressure, and we believe optimization is possible for pasteurization, preserving the blue color of the pigment. The reason that we were looking at this point is that we wanted to understand, is this decrease in color strictly related to the naturation of the protein that binds the pigment, or it is also affected by degradation by oxidation of the pigment itself. In order to verify and confirm this, we have extracted the pigment by itself and again used thermal and pressure treatments to test if we observed a loss in the absorbance. And in this case, we could not see, you can see here it in green, we could not see a difference in the absorbance between the pigment when it was cut from the protein itself. So the reason that we see a decrease in color of the pigment is actually the loss of the protein structure. So it was already suggested before, that's not something that we have invented, but it was important to verify that it is strictly related to the protein structure and not to the oxidation and degradation of the pigment itself. Next thing that I want to show you is the fact that pressure-induced changes in the structure of proteins can also affect the potential interactions of those proteins, of those plant proteins, with other small molecules. for us, we looked at polyphenolic compounds. So the first thing that is important to understand is the fact that application of pressure affects the surface hydrophobicity. And it is quite clear based on the images that I presented in the beginning, because upon application of pressure, we have an exposure of the inner part of the protein, and that's a main reason why hydrophobicity is expected to increase. And indeed, when we used rapeseed protein isolate, we observed 100% increase in the surface hydrophobicity upon application of 600 megapascals for 3 minutes. On the other hand, when we further treated the solution to 10 minutes, the surface hydrophobicity already decreased. And we believe this is the outcome of the beginning of aggregation of those components. It is not an easy task to understand which specific components begin to aggregate first, but that's something that we are working on currently. We could also see that this surface hydrophobicity is likely affecting the binding of small molecules to the protein itself. We could see a clear difference between the thermally treated and the pressure treated proteins in terms of binding with polyphenolic compounds. The information that a pressure can change the surface hydrophobicity was already shown also for a range of protein sources, lentils, peels, and faba bean. That is one of those conclusions that are overwhelmingly the same across a range of studies. But of course, the extent of the effect is strongly dependent on the protein source, and unfortunately, and that complicates significantly our research, on the purity of the isolate or extract that is studied. In addition, of course, as we have seen before, the pH of the solution is a very important parameter on the outcome of the impact of pressure when we are considering plant-based sources. Next step, it was unfortunately done so far only on animal-based protein, so it's ovalbumine from egg white, but it is a study that looked on the effect of a cosmotropic co-solute in the solution. So it's well known that small molecules like salt and also sugars can affect the behavior of proteins upon thermal treatment. And we wanted to see, is this also correct to the behavior of protein upon pressure treatments? So for that, we have tested ovalbumine at different temperatures and at different pressures in the presence of sucrose, a high concentration of sucrans, 1 molar. And we can see, of course, that this was done by CD spectroscopy, that upon application of both temperature and pressure, we see a clear change in the secondary structure of ovalbumine. That was quite expected. But if there is sucrose in the solution, it can only partially decrease the effect, the thermal effect upon the structure of ovalbumine, but it can fully inhibit the pressure effect on the structure of the ovalbumine. And we could also observe it in the particle size. So when we are looking at the particle size, there was a clear increase in the particle size of ovalbumine upon application of both pressure and heat treatment when no sugar was present, On the other hand, when 1 molar of sucrose was in the solution, the particle size did not increase at all after application of pressure, and on the other hand, it still significantly increased upon the application of heat treatment. And a similar conclusion could be reached when we are looking at the surface hydrophobicity. Both thermal and high pressure increase the surface hydrophobicity of albumin, but upon the presence of sucrose, the effect of pressure and temperature was lower, but more significantly was lower in the terms of pressure. The last thing that I want to mention about polyphenols and pressure and those interaction is the fact that pressure, because of the effects we observed on the interaction, will also have a clear effect on the bioaccessibility of those health promoting compounds. In this case, the study was done on kale, that is very rich in proteins, and with strawberry, that is very rich in anthocyanins. And we could easily see that thermal treatment significantly and negatively affects the bioaccessibility, while pressure has a significantly smaller effect. So to summarize, it is well known that high pressure processing affects different properties of food. It can be hardness and creaminess and viscosity and stiffness. But when we are thinking about effects that are not related strictly to the utilization of high pressure for food safety, we have other things to consider. The potential application of pressure can be in significantly different pressure levels and significantly different combination of pressure, temperature, and time profiles. Of course, we must keep in mind the source, the composition, and the way that we have extracted the plant protein sources. We should keep in mind the kinetics of those effects, especially if we are talking about gelation. It is a delicate balance between the kinetics of denaturation and the kinetics of aggregation. And we should not forget about how temperature, even in pressure-induced effects, can have a significant contributing effect to what we observe. At the end, pressure modifies protein structure, increases its surface hydrophobicity, and results in significantly different interactions with a range of compounds that differ from the effect that can be seen by temperature. And this paves the way to the potential utilization of high hydrostatic pressure, not just to ensure microbial safety, but also to assist in the development and in the engineering of novel plant-based solution. And with that, I will thank you for listening. Of course, again, thank the students that were doing the hard work and the different funding sources of those research activities. Thank you very much.
Thank you so much, Avi, for sharing your knowledge with us. So let's start the question and answer portion of the webinar. Remember that you can turn on your microphone to ask the question directly, or you can also write it in the chat and I'll read it for you. Any questions? Everything clear and sound? Comments?
Do you hear me? Can I ask a question?
Sure, go ahead, please.
I would like to ask Avi, how did you extract the protein? How the extraction method affect the result of these trials? Because we know we can extract the proteins in a lot of different ways.
Okay. Oh, thank you for the questions. In some of the proteins, the potato protein, it is a component used by the industry. So I did not extract it by myself. The rapeseed protein we extracted by ourselves. The reason that I state that it is clearly important because the way that we will extract will change the relative composition of the isolate. That is one of the main differences when in the conclusions that we can reach from proteins that are from animal-based. Very often the studies that were focusing on animal-based proteins were on single component purified one protein, while when we are talking about plant proteins, it is always a mixture, even in the purest isolates. And the way that we extract it will first affect the composition. So we clearly see different ratios of different proteins in different isolates. I did not show today, but if we have those results for P isolates, we clearly see different ratios between the proteins that will necessarily behave differently, just simply if we just consider the solubility. And secondly, it is the thermal history that also matters. So if we are talking about extraction and then drying that use a significant thermal effect, so we will already have partially or fully denatured protein, and our observation will be very much different if we compare it to, for example, proteins that were extracted without application of thermal treatment.
Yes, thank you.
Thanks for the question and the answer. Anyone else? Marco, do you want to open your microphone, please?
Yes, can you hear me?
Yes, go ahead.
Hi, thank you for the nice presentation, Abby. And I have a couple of questions. One is related to the slide where you showed that actually the bioactive was better preserved in the pressure obtained gel compared to the gel obtained by eating. I don't know if you have any explanation for that. And for the second question was related to reversibility. I don't know if you ever experienced during your research any case of vegetable-based protein that after a high-pressure treatment underwent a sort of partial denaturation and then refolded in its native state or in a different state that was not matching the native one. Thank you.
Okay, so first question. There is a very simple explanation for the effect that the vitamin C, the ascorbic acid, was better preserved in the high pressure gel. Vitamin C is very thermally liable. So to reach the same level of hardness of the gels between the thermal and the high pressure gels, I had to expose the solution of the protein with vitamin C for a significant thermal effect. And it is known that pressure has a significantly lower negative effect on the stability of vitamin C. So that's the reason. And actually this slide was actually the reason that we even started this research, because it was clear to us that we can better preserve compounds by application of pressure, but we wanted to make sure we can still get a similar strength of a hydrogel. So this is the question to your first point. The second point, I did not see or measure or quantify this semi unfolded state of plant proteins. But I have done the same for meal proteins, and it works. You can do it by heat. You can partially open it and then get it closed. That works. One of the problem is when we are talking about plant, and that was my last point, is that it is commonly said that, okay, it's an effect on the protein, but it's not a one single protein. It's usually dozens of different proteins in the solution. Each and every one of them has a different pressure or thermal stability. So it is a very complex interplay to find conditions that will result in the partial unfolding of all proteins. That will likely not happen. But indeed, we could see that there is a preference in the interaction to specific proteins. We saw it with rapeseed proteins, but we did not reach to measure it also after application of pressure, unfortunately. Not so far. But it is easier studied if I can purify a single component, and that's not an easy task. We have tried to do it.
Thank you.
Thanks so much. We have time for one last question. Okay, so, well, then, thank you so much, Avi, for this exciting talk, and thank you all of you for participate with us. Please keep on and check our website or Facebook page or Instagram account for more information, also LinkedIn. Regarding our next presentation, Avi, it was a pleasure to have you here. Thank you so much for this chat with you.
Thank you for having me, and I'm open for